Hello, everyone. I'm Rob Lindeman. I'm the Director of Clinical Operations at New South Wales Health Pathology, and I'm chairing this first important session. This session will showcase 4 of our early career researchers who will now give us a snapshot of their projects. The format is that the researchers will have a few minutes to present, and then there are questions, and you'll see that there is the opportunity to ask questions on the screen. So the first speaker is Jessica Agius, who's a postdoctoral research fellow with Public Health, New South Wales Pathology, based at Westmead ICPMR. So thanks very much, Jessica.
Thank you. I'm just going to share my screen now. Can everyone hear me? Oh, I guess you can.
Yeah, no, it's very lonely, but yes, we can hear you.
Okay. So hi everyone. Today I'm going to be presenting some of my research and my group's research on the dynamic nature of public health relevance of SARS-CoV-2 within host diversity. So before I begin, I'd like to acknowledge the Darug people, the traditional owners of the lands encompassing Western Sydney Local Health District, and pay my respect to elders past and present. So what are these within host variants or ISNVs? They are alternative alleles occurring above a given frequency threshold within an individual. And they can contribute to the functional characteristics of the virus, such as its phenotype and adaptive capabilities. So for example, we see here a person with a largely or highly homogeneous population of virus, as shown by the single color purple. Over time, minor variants can actually develop within this individual, as shown by the other colors of the viruses. Due to the transmission bottlenecks and dynamics of SARS-CoV-2, often the major variant, shown in purple, is transmitted, as you can see here. But other variants can also be transmitted, as shown here by these 3 individuals, resulting in a mixed population. More rarely, however, the minor variant, as shown here, can only be transmitted with no other mixed populations. So these are just some examples of the dynamics of transmission of SARS-CoV-2 and how it shapes its evolution. So the current research trends are looking at mutations in viral consensus genomes, and these represent the dominant variants. So these are known as the SNPs. But it's also very important to look beneath the consensus and look at the ISNVs, as new mutations can emerge within individuals. So looking at the ISNVs can actually show the person the full underlying within host diversity at the population level of that virus. So why are they important? There are a number of reasons why. They can provide early indications of diagnostic dropouts, particularly in PCR assays. They can predict the positive selection of mutations, so also predicting viral evolution over time. You can also help investigate drug resistance, such as those to monoclonal antibodies, and inform public health surveillance and case tracking. They can also uncover transmission dynamics and also advise on the design of therapeutics and vaccines. So there are a number of implications of ISNVs. They can overall increase viral virulence and exacerbate the patient's disease severity and clinical outcome. They can also lead to immune escape and also vaccine escape and treatment failure, so such as the development of drug resistance. So therefore, the aims of our research were overall to investigate and quantify the within-host variability in culture and clinical samples, so both severe and mild patients affected with SARS-CoV-2, using a probe capture approach and whole genome sequencing. So to the left here is a brief overview of our methods. So we did have a clinical cohort of mild patients, which were considered outpatients, and severe patients admitted to the ICU, and all patients were from New South Wales, Australia. Samples were collected between March 2020 and August 2021. We also incorporated culture replicates into our study, and these were from 3 patients from different lineages of virus. So we had lineage A, the Beta variant of concern, and the Delta variant of concern. And these were cultured from day 0 to 3, and we had various dilutions of these cultures. So basically, samples from these patients, RNA was extracted and subjected to a real-time PCR assay to confirm infection status and viral load. We then performed a viral enrichment step, which allowed us to enrich for and capture our virus, so SARS-CoV-2. We then performed whole genome sequencing and a number of bioinformatics approaches to obtain our variant profiles for each individual. So we called an ISNV, or the within-host variant, at a frequency of 0.05 to less than 0.9. And this will become more clear a bit later on. So this is just a visual representation of our clinical cohort. So you can just see here we had severe patients and mild patients with the shading, and different patients had different sample numbers. So we had longitudinal patients with multi-sampling and patients with only a single sample. But what's important to note is that we did exclude some samples and entire patients from our study because they did not reach our QC criteria cutoffs. This is an example of a variant profile, and for the purpose of time, I'm only going to be showing the spike region of the SARS-CoV-2 genome. And I'll just quickly go through a bit about the heat maps. So on the right in green, we have the patient identifiers, and the numbers in brackets just identify the symptom onset days. On the bottom here, we have a variant profile, so the nucleotide and amino acid variants. And then to the right here in blue, we have the frequency of the variants like a scale. So red indicates a SNP, and from orange to blue indicates a ISNV. So just going to the severe cohort over here, we had 16 patients with 26 samples, all had at least one ISNV, and there were approximately 4.2 per patient. So you can see here in the pink highlight for patient 105, there was 4 samples collected from day 7 to day 11 post-symptom onset. And you can see here that the variants aren't actually very consistent. One appears on day 10, disappears, and then we get 2 new variants occurring on day 11, these subvariants. So there's no consistent pattern here, which indicates their dynamic nature, particularly over time, over a patient's infection course. So to the right here, we have the mild cohort. We had 23 patients with 25 samples. The majority had ISNVs, and approximately 5.9 ISNVs on average per patient were identified. So on patient 495 here, we can see also 2 multi-samplings. You can see here the SNPs in red are consistent across each sample taken from the individual patient, but there is an ISNV or low frequency variant here, it disappears, and then another one turns up here. So you can just see that dynamic change in the individual over time. Finally, our culture replicates, you can see here, we had 3 patients with 33 samples, and all at least had 1 ISNV. So this first kind of bit here is the lineage A, and you can see here where the blue highlights are that we never actually reached a SNP, which is indicated by red. We just had consistent populations of ISNVs changing frequency over time and dilutions. So here you can see this is the days in the second bit of this nomenclature and the dilutions. So they don't actually remain consistent, always changing the frequencies. And then in this beta variant of concern patient, we do see an ISNV occur here, a very low frequency one indicated by blue, and then it actually flips to a SNP, so a higher frequency variant later on during the course of culture. So to the left here is just a column graph showing the counts of ISNVs depending on the different genes across the SARS-CoV-2 genome. At the top here, we have culture in blue, and then the clinical cohort in the bottom. So severe is in red and mild is in green. And where the yellow highlights are, they just show that the majority of ISNVs actually occurred in the spike gene in both the mild and the culture cohorts, which is interesting because the spike is obviously very important for gaining infection in the body and for vaccine targets. So the right here is just a box plot showing the range of frequencies that we obtained with our ISNVs, depending on the different genes in the SARS-CoV-2 genome. And again, spike and culture had the greatest range of frequencies of ISNVs. And there was actually a significant difference in the severe and mild cohorts. So severe had a higher average of frequency compared to the mild cohort, indicating that they were more likely to actually flip to a SNP potentially. So we also wanted to look at the genomic clusters. So at the start here, we had 33.1, which just shows that we had 2 household contacts, which are indicated by the diamonds. And you can see here that they actually had identical SNP profiles where the other individuals in that household cluster did not have these. So this indicates clearly linked transmission events. And again, here for cluster 17.5, we have 3 household contacts, and they all have exactly the same identical profiles. And that can also show us and also help epidemiologists link these transmission events occurring. So we also wanted to look at mutation ratios. Basically, this KAKS ratio just determines whether the mutations gained are beneficial, neutral, or purifying. And because we had values greater than 1, it indicated that our selection was positive, and these mutations gained by the genomes in our study were likely beneficial to the virus. So in conclusion, our results did demonstrate significant heterogeneity and dynamic frequencies of these ISNV profiles across both the clinical and culture cohorts. And as you saw the different dynamic frequencies occurring across multi-samples of the same patient, this indicates that there is probably likely selective pressure occurring on the virus. The SNPs in cultures and clinical samples remain relatively consistent across the days. And ISNVs and SNPs were shared within genomic clusters as evident by the identical profiles. So this does clearly show linked transmission events occurring. So they are important. They can predict clinical outcome, these ISNVs. They can help us understand hospitalization risk. They can help also uncover viral transmissibility and traceability, and they can help in the design and development of vaccines and other effective therapeutics. So thank you. If you need to contact me for any questions, I'm happy to answer. There's my email. I'd like to thank everyone on the team, and especially the people in the photo. That's our team there at Sydney Public Health, and also New South Wales Health Pathology. So thank you so much. Any questions, I guess?
No. Thanks very much. Oh. Trying to turn my video on, but doesn't want to do it.
There we go. I think it's working.
There I am on. Yeah, I'm clicking too often. So no, thanks very much, Jessica. Now we've got 57 people on. There must be people with a question. Don't forget to ask questions. I can see when people do. So there's clearly quite a lot of variability in severity of SARS-CoV-2. How much of that variability do you think relates to viral factors and how much to host factors? Is this the major player in the differences between severity, or was it mainly host factors?
We didn't look potentially into the actual causal effects of why we had the diversity, but because what's known is obviously host factors play a major role, depending, especially if you're immunocompromised. People that have these SARS-CoV-2 infections over a long period of time often are the ones that do develop these minority variants, and they are mostly probably, I think, likely the cause of the recombinants that we're seeing now, because the virus has been exposed to the host over so much time that it can actually develop these new variants and successfully transmit them to susceptible populations. But obviously, the virus is also evolving, so it's basically accumulation of both the viral and host factors. So don't know to what extent, but I think it definitely depends on the host and also the variant that's actually circulating.
Thank you. And does the variation, did you look at vaccination history and types of vaccinations? Does that influence the emergence of some of these variants?
I definitely do think, because vaccination is a player on how it actually it causes, what's the word, what I just said? It does put selective pressure on the virus to evolve. We did have vaccination status, but we didn't report that in our paper. Basically, I don't know if we had ethics for it, but we didn't. I don't know the results off the top of my head about vaccination status, but definitely vaccinations does put that selective pressure on the virus and to obviously evolve over time. So,
yeah, and then do people who've had different vaccines behave differently to people who've had the same vaccine every time, or I guess you don't know that?
No, I don't. We didn't look into that specifically. No, sorry, but that is interesting to look into in the future.
Okay.
Thank you.
All right. Well, thanks very much, Jessica. No one's asking any questions here. Either that, or I've not been able to master the technology.
I think you did a good job.
Thank you very much. Okay. Thanks, Jessica.
Thank
Okay. Good.
Okay.
So the next talk is given by Greg Walker, who's a research associate with the Virology Research Laboratory at Prince of Wales Hospital. And we're running 1 minute ahead. So hopefully Greg has turned up. Otherwise, I'll be ad-libbing for a little bit. And there he is.
Hey, Rob. I'm just trying to share my screen. So it's saying the host has disabled participant screen sharing.
We can see you clearly, but we can't see you talk. I'm glad I'm not the only one.
There we go. It's coming up now. So is that visible, Rob?
Yep, all good. Thank you.
Is it the full screen or is it the
So
presenter?
we're seeing the title page, Research
Okay.
Forum.
Okay, that's great. So hi everyone, thanks for joining. So today I'll be presenting some of our group's work on the rapid evaluation of COVID-19 monoclonals to guide clinical use. So for those, I guess, unfamiliar with this type of therapeutic, I'll just give a quick introduction to monoclonal antibodies. Antibodies are essentially antigen-binding proteins that are produced by immune cells of the body, specifically B cells. And B cells can be isolated in the lab and cloned to produce large quantities of therapeutic monoclonal antibodies. And these monoclonal antibodies bind to a single and specific target. Think of it, I guess, like a lock and a key. And monoclonal antibodies are given intravenously and can be used for the treatment of infection or also the prevention of infection for at-risk populations in situations where vaccines or antivirals might be considered unsuitable. So, for example, an immunocompromised patient who fails to mount a high immune response to a vaccine. And monoclonal antibodies for COVID-19, they work by binding to the spike protein of SARS-CoV-2, directly, I guess, blocking or inhibiting ACE2 receptor-mediated viral entry. And there are a few other monoclonal antibodies that have different mechanisms of action as well. And since COVID-19 monoclonals bind the spike protein, the emergence of SARS-CoV-2 variants has really challenged their efficacy. So since early 2021, we're seeing an accumulation of mutations in the spike protein, and particularly within the receptor binding domain of the spike protein. And in this region, I guess, you can see highlighted by the red in the figure. So that region of the spike protein is where many of the monoclonals that are clinically used actually bind. Not all of them, but most of them. So rapid evaluation of monoclonals against new viral variants is needed, I guess, to determine the efficacy of clinically used monoclonal antibodies. So this is a list of monoclonals that have regulatory approval. So there's 5 that have received emergency use authorization by the FDA in the US, and the top 3 on that list there also have TGA approval for use in Australia. So how do we determine monoclonal antibody efficacy against new variants? I guess the best data that we can have for this is in vivo data. So that could come from animal models, it could come from human clinical trials, and better yet, real-world clinical evidence. I guess the only issue with this type of data is the time it takes for these types of studies to be planned and for the data to be collected and published and disseminated. Quite often, by the time this information comes out, it's no longer relevant because there's new variants circulating within the community. So in vitro data really can give valuable guidance within quite a short time frame. So we've established a collaborative pipeline to enable this rapid evaluation. At New South Wales Health Pathology Randwick in the Savard laboratories, we have a COVID-19 surveillance team. These guys have been performing next generation sequencing of viral genomes since the beginning of the pandemic, and they've been identifying new variants as they emerge in Australia. And soon after identifying a new variant, we'll take the virus to the PC3 facility at the Kirby Institute, who we collaborate with quite closely with Stuart Turville. And we'll grow up viral stocks of these new variants, and these lab stocks are used for live virus assays in vitro. So this slide essentially just shows us what a neutralization assay looks like in the lab. Essentially, just live virus is mixed with the monoclonal antibody at various dilutions, and then it's added to cell culture, with neutralization calculated as a measure of cell viability. So a strongly neutralizing or a monoclonal antibody at a high concentration, such as we see on the left there, provides protection from infection, and we have healthy, viable cells. And as we move to the right, at lower concentrations, you essentially have increased cell death, lower cell viability. And we measure this using some image analysis software that really automates it and streamlines it for us. So this pipeline was used really effectively with the emergence of the Omicron variant last year. So Omicron was first identified in late November, and a few days thereafter, it was classified as a variant of concern by the WHO. And our surveillance team actually detected an Omicron case in New South Wales within a few days of the variant appearing. We actually had data available on these monoclonals within about 2.5 weeks from initially identifying the variant. And we found that many of the clinically used monoclonals either completely or significantly lost activity against the Omicron variant. And this data was cited by the CDC in their updated guidelines for COVID-19 treatment following the emergence of this variant. And I believe within a few weeks of this updated CDC guideline, the FDA actually rescinded some of their emergency use authorizations for a couple of these monoclonals. So this work has continued throughout 2022 with the emergence of the Omicron sublineages. And we won't go into the data too much, but we see varying efficacy of these monoclonals against new variants. And this, I guess, highlights the need for continual therapeutic evaluation and monitoring. So obviously the circulation of variants is quite dynamic. BA5 has really dominated since the middle of the year, but in this figure, this is from the CDC variant tracker. You can see over the past few weeks, there are a number of new variants that are emerging and will likely supplant the dominant BA5 fairly soon. So I also just wanted to, I guess, highlight the need to follow up the lab data with in vivo studies. So like I mentioned earlier, there's a bit of lag in dissemination of in vivo data and clinical evidence, but there are some studies becoming available now from the earlier BA1, BA2 waves, which occurred much earlier in the year, probably around March or April. And what they show is essentially that a reduction in neutralization in vitro doesn't always equate to a loss of clinical efficacy. So for example, sotrovimab actually lost its emergency use authorization after in vitro studies found about a 50-fold reduction in potency in the lab against the BA2 variant. But there are some studies that are coming out now, a small one in the UK, well, 2 small ones in the UK actually, that found essentially no differences in outcomes between BA1 and BA2 infected patients who received this monoclonal antibody. So I guess just to wrap up, therapeutic efficacy varies with the continual evolution of the virus. So ongoing evaluation is needed in vitro against new variants and also in vivo to follow up this lab work. And there are updated treatments on the way. So there's a number of broad spectrum monoclonal antibodies that have been developed, and they maintain neutralization against all variants. And there's also another class of monoclonal antibodies that we haven't spoken about today that are aimed at controlling inflammation and later stage COVID symptoms. So thank you all. I guess, like to give a quick shout out to the members in the Savard Lab and at the Kirby Institute, who we collaborate with very closely. And a lot of this work was performed during my PhD, so also to my supervisors, Sasha Stelzebrade and Bill Rawlinson. Thanks.
Thanks very much, Greg. Now, look, so audience is very light on for questions. I zero. The only questions I'm getting are from Stephen Bro, who's gone old school and is texting me questions. The first of which relates to the, I guess, cross-reactivity between these monoclonal antibodies and other targets, and I guess potential for side effects. Presumably the time to development of these and the need to bring them in quickly limits the amount of testing that can be done. Can you actually predict specificity or otherwise of these targets?
So I guess in terms of the safety, monoclonal antibodies, they don't have a long history in infectious diseases, but they have been used over the last 20 or so years for RSV. So palivizumab is used in infants, particularly preterm births. And it has shown in those instances a really good safety profile. And I think it has got a really good safety profile. The SARS monoclonal antibodies have shown since their introduction a really good safety profile as well. Yeah, I guess it's quite a dynamic situation, and they've figured it out as it's been rolled out essentially. Yeah, does that help?
Yep. And so, I mean, this is a battle we're never going to win, but it sounds like. What's the time to development? I mean, from identifying a monoclonal with specificity, what's the time to development of a commercially, a commercially available monoclonal?
So some of the earliest really big papers published in journals like Nature, for example, those really appeared in, I think, around July 2020. The big papers about antibody discovery. And by the time these were rolled out commercially, I think the earliest ones in the USA were in February the following year. So probably 8 months, I would guess, from discovery through to commercialization. But those products weren't available in Australia until the end of 2021. So yeah. Yeah. Okay.
Now we're starting to get a few questions in, so that's good. First question is from Sean Roman. In your lab assay, how much monoclonal antibody did you use and what viral counts were used? And how does that sort of relate, I guess, to clinical practice? In other words, to the viral load use on a patient.
Okay, so we usually start the monoclonal at a highest concentration of 10 micrograms per mil, and then we do a 1 in 3 dilution series essentially. And we see the IC50 values for the most potent monoclonals. They come out, in the single digit nanograms, in 5 to 10 nanograms per mil. But I guess how that relates to, I guess, the dose received in patients. I think the clinical dose far exceeds the dosage, the IC50s that we see in the lab. So like the sotrovimab example I gave just in the talk, in the lab, we saw a 50-fold reduction in neutralization potency. But then in patients, there was essentially no difference.
Yep. Another couple of questions, and we're going to run out of time before we run out of questions. Is approval ever given based on just on in vivo results? In other words, clinical trials rather than your laboratory finding of efficacy?
So I think approval for all of these initial approval by the FDA and the TGA for these products wasn't given until there was clinical trial evidence. Yeah.
And then Emmanuel Favalora has asked a question. There's a humanized monoclonals. Does efficacy fall in an individual over time?
Yeah, so I don't know the exact numbers, but there is a gradient of bioavailability. So particularly for, I guess, Evershield is used prophylactically, and there is a waning over time, much like you'd have with a vaccine antibody response. Over time, the bioavailability goes down, and it probably needs to be topped up, but I don't know the exact numbers.
Yep. And the last question is a question you can't answer, which Bente's asked. With the new variants coming and being resistant to antibodies currently approved, how are we going to stay on top of this? I don't know if there's an answer to that, but you can give it a go.
Yeah, it's getting difficult because there's so many variants at the moment. In the lab at the moment, we're growing 20 different branches essentially of the Omicron sublineages. So I don't know moving forward exactly how we'll stay on.
It's like the sources of apprentice. Now, thanks very much, Greg. We'd better move on to the next talk.
Yeah, thank you.
Thank you. So the next talk is from Chris. It sounds like this is all COVID stuff. Next talk is Chris Cott. So I think many of you will know Chris, who's been working with the point of care team at New South Wales Health Pathology. So Chris, would you like to take over the screen?
Not a problem. Thank you, Rob, and my previous presenters. Today, I'm going to dial it back down a little bit and go into something a little bit light and fluffy. I'm going to go into rapid antigen tests. So, and it's a small-scale assessment that was conducted in order to basically work out what's causing some of the false positive issues at the beginning of the year. So I'm not going to spend too much time on the background. You all know that with the introduction of new tests, there's always some drawbacks, or I should say new/old tests in rapid antigen. And we all are very much aware of their performance limitations now. We all know that introductions of new tests carries lots of ongoing challenges. And I'm not sure, can you see this diagram on your right, or am I?
Yeah, no, that's good. No.
Okay,
can see a bit of it, not all of it. Yeah, that's fine now. Yeah,
there
that's
you
good.
Go. Okay, so apologies for that. So basically, I'm sure most of you in the audience are aware of how a lateral flow device works. You know, you've got an analyte, you know, you've got an antibody conjugate or target, which will bind to it, you know, as it draws across the lateral flow device. If it's carrying the virus, it's going to be picked up by the test antibody. If it's not, it's going to be picked up on the control band. And I guess diagram 3 shows that where you've got both occurring. And now I'm going to jump into what New South Wales Health hypothesised based on the manufacturing of the rapid antigen kits, which utilise a antibody which has been buffered. We hypothesised that the introduction of an acid environment would lead to some false positives. And that's also because we know that they work in high affinity at a neutral pH, somewhere between 6 and 7. So let's have a look at the timeline, because this was quite a rapid assessment conducted at the beginning of the year. So as history records, from the 1st of November 2021, you were allowed to use rapid antigens in home, but there pretty much was no supply until around Christmas time. Entering into the market, also saliva kits were around. And hold that in your mind, because that becomes a little bit more important later in the presentation. And this is my shameless cross promotion. And the reason saliva became more known is you can see, using the school's rapid antigen feasibility trial, why the community preferred saliva. So sorry about the shameless cross promotion. This assessment was conducted in the end of January, and basically it came through the Ministry of Health and the TGA, because they were under pressure to try and work out what was causing the false positives. And we had basically a week to try and work it out. So as always, a lit review is the beginning point, and leave it to that next generation who had already worked out some of the problems. So as school kids will do, They are after a day off, so they figure out how to get a false positive. So along the way, we had to work out whether this was true for the Australian context and what mitigation strategies could be put in place to try and overcome this. So the methodology that was created for this small scale, we utilised 7 different rapid antigen products. They're a mixture of 3 saliva, 4 nasal, and we used 6 different solutions added to those test kits. We also did a control band using a pH curve, which was basically titrated using 1 molar HCl, where we could see the different effects over the different pH range. We measured time intervals as well. So just for your reference, these are the kits that we used and also the solutions that were added. So you've got vinegar, which is down the bottom at your pH of 2 to 3, and then your glucose tolerance is around 6 to 7, keeping in mind that that is around the target affinity level for rapid antigen kits. Okay, so what do we see? On these graphs, you will only see the positive outcome results. Negatives are not displayed. So time intervals, 30 minutes, 60 minutes, and at 100% buffer or the normal kit, there were no positives seen. So as you would expect, vinegar, the most acidic, generated the most false positive results. Substances like wine and orange juice were heavily affected on the salivary kits, particularly at time interval 0. And across the board here, we see that when the buffer was replaced with the actual by greater than 50%, we got false positives. And that's consistent across the board here as well. So false positives occurring when the buffer's been replaced, and also saliva generating high levels of false positives. Interesting on this that mouthwash didn't produce any false positives on the nasal variety of kits, and saliva didn't produce any on oral glucose tolerance solution. But if you remember back to oral glucose tolerance solution works at a pH between 6 and 7, which is around that target range. So just looking at the control band, so we've got the pH scale here. We're seeing false positives occur anywhere from under a pH of 3.9, and a number of false positives occur in the range between 4 to 6. Rapid antigen kits, when they were run at a pH of 7, didn't generate any false positives, and obviously when no normal buffer was applied to the kits, we didn't see any false positives either. The interesting thing to note about the effects on the pH scale, those kits which produced a false positive around between the 4 to 6 mark were also the kits which was strongly associated with producing false positives throughout the additions of various solutions. Say, for example, the wine or the orange juice, etc. These kits were the ones that also produced it. And I'm going to jump pretty quickly into the discussion in the interest of time. And I'm not going to spend too much on this because I really want to get into the next slide. But basically, you can see across the board that false positives are quite possible to occur on both nasal and saliva variety of rapid antigen kits. And false positives could potentially occur if any of these solutions were consumed and testing began immediately after. This brings in to sheds light that clinicians need to be careful or people need to be careful when taking a rapid antigen kit if they've just consumed a solution which is of this acidic variety. But how and why does this occur? Why are we seeing these artifacts? So just bear with me, we'll go on a little bit of a journey and you should know by the end. So many rapid antigen kits have been licensed for point of care for SARS-CoV-2 detection, and they vary in their methodology, test frequency, and settings on how to use them. Rapid antigens are well known to be less sensitive and specific than PCR, and false positives have been known to occur in low disease prevalence areas. Direct sampling produced false positive results, which were visibly similar to that of true positive results seen on true COVID patients. So basically, you couldn't tell the difference between a false positive or a true positive, particularly when solutions like vinegar are applied, which don't leave any streaking line on the kit. The likely explanation for these false positives is non-specific interaction, which is occurring between the solution and the capturing antibody. Since pH generates false positive results, it is proposed that pH alters the conformational structure and there is non-specific binding occurring. This study was able to show some mitigation strategies, which is basically ionic strength, time intervals, buffering capability, the addition of buffers, etc., could potentially reduce the risks of false positives being generated. Typically, false positives arose from cross reactions or interfering substances. The mechanism by which false positives are generated are related to non-specific interactions between the antibodies and the surface substance. If we examine the manufacturing process, this will shed some light. So basically, they have a negative charge reducing agent, which is attaching the capturing antibody to the test strip. When the pH is acidic or less than the isoelectric point of this criterion, the negative charge then binds to the non-specific positively charged protein, and therefore you end up with a false positive occurring on, or the capturing antibody basically having a conformational change and triggering a false positive. Other studies have demonstrated that by addition of proteinase K can abrogate this phenomenon. There are other ways too, such as the addition of buffers and other factors like that, which can also remove the false positive. Some future directions, we'd like to increase the size of this test, go into a full patient trial and add in other variables, which could also be triggering false positives, such as location, time, altitude, health status, etc. So that's probably my take-home message is basically don't eat or drink before doing your app. And any questions?
Thank you, Chris. Yeah, it was an interesting.
Sorry for racing through.
No, that's all right. That's okay. We're good. I think we can see a very busy screen. So looking for questions from the audience. It worries me that your white wine and vinegar are so alike in composition. They're only a pH point apart. So what? But quite.
Are you suggesting my wine was no good, Rob?
Well, possibly. So I guess the serious question though is obviously there has been time pressure. And TGA registration is sort of, I guess, what we use as the, stamp of performance of a lot of these devices. What's the? And it's interesting that all these UK school kids figured it all out so early on. What's the mechanism for the TGA assessing these sorts of devices before they become available? Because I mean, finding out all this stuff after they're available does seem a little out after the event. Well,
as I'm sure you're aware, Robert, it's assessing it after the cart is basically bolted, isn't it? So it's a horse before the cart scenario. So a lot of the TGA review relies upon other such as FDA or other approvals that the companies or devices may have. And there's a lot of literature review by the TGA and basically relying upon the information presented by the manufacturers. So delving into what they've produced.
Thank you. So the next question, there are a lot of questions now. So Sean Roman would like to know how much wine and orange juice and vinegar do you need to drink to get to sufficiently lower the pH? I mean, obviously you're not going to stick these in neat vinegar.
so obviously, Sean, what, day 2 on the job and you're after time off already, Sean?
That's right.
It scared you that badly. So basically, as you can imagine, it's about altering your pH levels. So each individual is going to be slightly different to the amount, and whether they swirl the wine or whether they're drinking it straight. But once you've created alterations in the pH, then that's when you're likely to see that false positive arrive, particularly if you've consumed that beverage and then testing straight away.
Yep. And Michael Wiley, I guess it's a comment rather than a question, but it shows the key to the reliability of these and other point of care results is dependent on pre-analytical issues rather than just the performance of the test. And I guess our approval pathways evaluate the performance of the tests, but maybe don't take into account the pre-analytical.
Absolutely, and that could be probably said too for the laboratory side of things. We just don't see it as much. So it's true for all pathology tests, how important pre-analytical is.
Yep. And I'll squeeze one last one in. And again, it's another one of those Dorothy Dix ones. Is the lack of interfering substance tested an artifact of many tests being granted emergency use authorization with a lack of comprehensive analytical data? And if so, will these tests need to submit full analytical data to remain registered tests? And I think that's absolutely true, isn't it?
I can't speak for the TGA, of course, but yeah, obviously that seems like something that would be very prudent, doesn't it, Rob?
Yeah, I mean, there's a whole lot of stuff that happened very, very fast that we'll need to review. But anyway, look, thanks. We're out of time. So thank you very much, Chris, for an interesting talk.
And thank you all for listening.
So the last in this session is, again, unsurprisingly, to do with COVID. This is Jessica Johnson, who's also research fellow at ICPMR Westmead. And Chris's name's still on the screen, but Jessica's appearing right now. Thanks very much, Jess.
Thank you. Sorry, just forgot to unmute
Yep.
there for a second.
No, you're on. Good.
Okay, so today,
You're on mute.
am I still?
No, I can hear you very clearly.
Sorry. So today I'm just going to talk a little bit about our group's work using respiratory viral oligo panels to identify potential co-infections in SARS-CoV-2 positive patients. So the ability to detect these co-infections is often limited due to an over-reliance on specific PCR assays and amplification-based sequencing protocols, especially at the beginning of the pandemic when the push was to generate really high-quality genomes that would give us the resolution to detect transmission events and to cluster outbreaks. After the tipping point with vaccinations, when this kind of resolution became less critical to mitigation, public health containment strategies were easing, and it was expected that there would be an increase in high burden respiratory pathogens, for which the capacity to conduct these kind of genomic surveillances were and still remain quite limited. So we moved our focus from just getting these high quality SARS-CoV-2 genomes to being able to detect potential co-infections with these other pathogens. So RVOP, it's a probe-based capture method, which means that it requires a lower specificity to the target of interest, allowing
Jessica,
for
you're still
detection.
on the openings. You're still on the opening slide. Are you hoping to advance them?
Yeah.
Okay.
No, I'm good. Sorry. So it allows for the detection of these 42 distinct respiratory pathogens in one sample. So I'm just going to talk about its application in kind of 3 different ways. But first, very quickly, the workflow looks like this. We start with those very unpleasant nose swabs that we all experienced. We extract RNA from those. We create single and then double-stranded cDNA. It's fragmented and then uniquely tagged. It then becomes PCR amplified, pooled, and then the libraries are hybridized with capture probes. These are then amplified and we sequence with a target of 1 million reads per specimen. So we do a bit of a deeper kind of sequence, and this is to increase our chance of getting very high coverage. So in this study, we kind of did a 3-tier way. First, we spiked positive SARS-CoV-2 samples with influenza A, RSV, and human adenovirus, which was just as a proof of concept to simulate a mixed viral infection. So we then sequenced the samples to determine the sensitivity of the protocol. Secondly, we looked at its applications in kind of surveillance. So we looked at samples that were being routinely sequenced for SARS-CoV-2 for the possibility that they had co-infections that were being missed. And then finally, we looked at a specific case study. So going from proof of concept to just kind of routine to an individual that we wanted to get a closer look at, an 11-month-old patient that was known to be PCR positive for both influenza A and SARS-CoV-2. So for the spiking experiment, our proof of concept, we found that the samples that had medium and high viral loads returned genomes with a percent coverage above 90, which is quite good. And that as the CT or cycle threshold went higher to roughly 30, which is a low viral load, the percentage began to drop off. And this is consistent with other target-specific methods, like our amplicon-based. But with RVOP, you can achieve this kind of higher resolution in terms of population specifics. For instance, we know that it's flu A, H3N2, and this is important because knowing this kind of information can help in determining treatment options or seasonal vaccine targets. So next, when we looked at the samples that we had already sequenced, we found that 12% of those cases had previously undetected co-infections, including metapneumovirus, polyomaviruses, and another case of a flu and COVID infection. So that's quite high when you're talking about 12% of, just our 33 that we looked at. For the case study, initial targeted sequencing found that the SARS-CoV-2 was an Omicron variant. So this is our amplicon-based sequencing. But because we knew that they were PCR positive for flu A, we wanted to look at getting more resolution onto the flu A and potential other pathogens. So because we were interested in these co-infections, we decided to sequence it again with RVOP. So from that, we found that the majority of the reads were from a different coronavirus altogether. They were from coronavirus 229E. It's one of the 4 common coronaviruses known to cause mild disease, but it can be quite severe in infants. And remember, this is a 11th-month-old. We also got a complete SARS-CoV-2 genome from less than 1% of our reads. So remember, our target is about 1 million reads. And we also found reads to influenza A, KI polyomavirus, other common respiratory virus in children. Not enough reads to create a complete genome. So we know that this child had kind of quite a variety of different infections. So from our proof of concept, we know that it can produce high quality SARS-CoV-2 genomes, but it can also produce complete or near complete genomes of other common viruses with a high resolution. From our routine sequencing, we can see that we're missing the detection of co-infections and identification of other pathogens that are in circulation currently by focusing just on SARS-CoV-2. And we know that if we use this kind of panel, we can find potentially more viruses or other viruses that are infecting ill patients that we wouldn't be able to find because we either don't have a PCR test for them or we're simply not looking. So in conclusion, the addition of this kind of panel-based sequencing method could offer public health laboratories a really useful tool, both for surveillance of kind of known pathogens and for picking up emerging pathogens that we don't have testing for currently. So if you've got any questions at all, I'm open. So thank you for watching, and thank you to everybody else that helped on this project.
Thanks very much. Thanks, Jessica. That's very interesting stuff. So there was a question that appeared and then disappeared from Stephen Bray. So Stephen's asked, is this infection and colonization? Is it actually infection, or is it just colonization? You're detecting stuff that happens to be there in the background.
It can be a little bit of both. So a lot of it we go off of percentage. So if it doesn't reach a certain percentage of the total reads, we consider that to be background noise. So it might have been a bit of a hit or a miss, or it might have just been there. And like the higher the percentage of reads that it is, the more likely that it is like a proper infection as opposed to just a colonization. So for that particular child, they definitely had SARS, and they definitely had the other coronavirus, and they may have had a little bit of colonization with those other viruses.
Yep, thanks. Another question from Sean: how susceptible is the application to expanding areas? Does this hinder detecting variants? And I guess, is the amplification faithful?
Yes, so it's quite good we get high coverage and complete for just COVID genomes. And so we can identify all the SNPs and put it into their variants, into their lineages. And in fact, it's better than amplicon based specifically, like your Arctic, because if you have mutations that happen in primer sites for Arctic or COVID specific, lineage specific amplification, then you're going to get dropout and you're going to lose that coverage. Whereas because this is not as specific, it will kind of much better at going with the bumps in the road. And I want to also mention that this is a specific kind of viral panel, but there are other panels that can be applied to different kind of areas. So there's another panel that we currently use that picked up monkeypox and produced better monkeypox genomes than just kind of metagenomics and throwing all the reads possible at it. So it's more cost effective as well.
Yeah. So is this the sort of thing that you should be doing on an ongoing basis as a public health activity, just to monitor what's going on in the community? Is that do you see this as sort of a routine public health activity?
I think it definitely could be, especially if routine sequencing is something that your laboratory already does. It's the same kind of price point to do a panel as it is to do specific sequencing, especially if you can, up the number of samples into it. So we're not talking like way more, but you get more data out of it.
Yep. So another question from Greg Walker. And this is about the panel you're using. So are these all respiratory viruses? And sort of how many viruses does it detect?
It can detect up to 42 different ones, and it can be kind of strain specific as well, if that's what you're interested in. So it can detect different varieties of flu, for instance, and different varieties of other things. But this particular panel is 42 different viruses.
Yep. And question from Stephen Bray. So you obviously tested people with infections, but have you tested healthy controls to see what the background is?
Yeah, so we did spiking on positive, and we did spiking on negative. So we know, like COVID negative, we did spiking into it with the different ones, and the controls worked like the controls were meant to work. And it's also where we kind of get that threshold of, do we believe this is actually real? And based on the kind of the number of reads, because
yep.
in laboratories, you're going to have stuff floating around.
So that's
anyways.
How you pick the threshold. Yep. Okay.
Yep.
And another question: have you performed cost benefit and workflow turnaround time comparisons to standard PCR respiratory multiplex testing? Because it sounds very labor intensive. This comes from Simon.
So we haven't compared to just PCR multiplexing, because it's a different tool. This is like the sequencing equivalent of multiplexing. Compared to doing like a amplicon base, so we're only targeting one pathogen within it, the turnaround time for these methods is pretty similar to each other. It's equivalent. protocol is slightly different. If you're talking about standard kind of Illumina sequencing, if we're going into ION where you can do that really quick turnaround, it'll be different. And I haven't done a detailed cost analysis. It's just kind of stuff that we figured out over the 2 years.
Sounds expensive. Okay, we better stop there. We're right on 2 o'clock. So thanks very much, Jessica. And thanks to all the speakers for an excellent session. A lot of COVID. What will we do without COVID? So there's a 20-minute break. But before you go, there is a facility to vote for your favourite talk of the ones we've heard. And next session starts at 2:20. So thanks very much.